floor is yours.
Okay, so good afternoon and good morning for those in the Caribbean. So today I would like to introduce you Samuel Bernades, who I had the pleasure to welcome in Berlin while I was doing a postdoc there at the Museum of Natural History. So he was starting his PhD, and now 3 years or 4 years, 4 years has passed.
3 years.
And 3 years.
Yeah.
It comes quite fast.
Yeah.
So so now you're finishing your PhD, and so before coming to Berlin, you were a student at the Federal University of Minas Gerais in Brazil, and then you went to Bangor University in the UK, and so now you're in the museum in Berlin, and you're planning to go to do a postdoc in Bielefeld. So that's what you said, right?
Yeah.
Okay. So Sam started his PhD in the team of Thomas and Christina von Rintelen, where I was also doing my postdoc on a related subject with the Atyidae shrimps, so freshwater shrimps, especially from Southeast Asia. And now Sam is working on their biogeography based on the phylogeography. So yeah, I leave the stage to you now.
All right. So this is the final chapter of my thesis. So the results that I'm going to present here are not published, and there are a few problems with them that we still have to sort out. Mostly is because there were changes in the taxonomy, and we need to change the outgroup, and then we had to extend the project a little bit. But I stand for all the results, for most of the results that I'm going to show here. And when I not, I will definitely say so. So I will start by giving you a short introduction about the family I work with. The Atyidae are a family of freshwater shrimps with over 500 species in almost 50 genera. I think the latest revision showed 46 valid genera. The taxonomic problem inside the family is evidenced by the fact that over 70% of all of its diversity is concentrated in one single genus, which is Caridina. And this did not stop the taxonomy from growing. We have about 10 new species described per year for the past 10 years, and over half of them only consider morphological apomorphies and similarities, some of which have been defined decades ago and are known to be problematic. The result of that is that we have many species complexes. It could also be that the high number of species complexes and the high number of species inside one genus could be due to a high recent speciation rate or a past speciation peak followed by low extinction rates. But to say anything about evolutionary or to make inferences about evolutionary causes for all this, we need to date this. We need calibrate the time of this evolution. And that's another problem with the Atyidae. The Atyidae has a very poor fossil record, and the oldest fossil is in the Cretaceous, but the fossil taxonomy is also very unstable. The most inclusive work with the Atyidae so far is from 2012 by Christina and collaborators. Valentin has a few other works that are very inclusive as well, but the most inclusive are from 2012 by Christina von Rintelen. Here in this work, I will focus on this group, the Caridina group, because it contains most of the diversity and it contains all of the Caridina species. In the work by Christina von Rintelen, they recovered these 5 groups, and they proved that all the subfamilies were invalid. So this is one of the suggestions, is to treat these 5 groups as subfamilies. But again, we need a more robust analysis. And here I come in. I come in with a genomic analysis to actually add to more data to these relationships. This is an excerpt of their tree containing only the Caridina group, And you can see the Caridina marked in red along the tree. And you can notice that Caridina is not only paraphyletic, but it's polyphyletic. It's spread all over the tree. It has very confusing relationships. It's not a valid genus at all, unless everything is Caridina. Christina's work also showed that this group, studying the phylogeny of this group, is very sensitive to fossil calibration schemes. They had several schemes, they tried several schemes, and the dates changed a lot. They had only one fossil calibration. They used, the other schemes used an external fossil calibration from a sister group and extrapolated date from a previous work. The problem with the fossil calibration that they use is that they use this fossil here, Atyidae hoxoi, pardon me, and this Atyidae is an extant genus, and this already shows the problem with using this fossil. But if that's not enough, in 1991 this fossil changed to another family and another genus, and it was now known as Pseudocaridinella. But of course, it was not satisfactory enough. And in 2019, it changed again to another genus, again inside Palemonidae, another family, to Baiacaris. So you see that the only fossil that we have now that's actually useful for calibrating is Deoclosia martinelli, which is assigned as a stem group Atyidae. We have other fossils, but most of the fossils, they are very recent. Some of them are contained in extant genus, which makes their classification very hard. And most of them are Caridina, which also makes the classification very hard because the genus itself is not very reliable. But Deoclosia as a stem group, Atyidae, makes calibration very hard because stem group depend on the outgroup as well to be calibrated. We need to calibrate on the root and not on the in-group. And this makes calibration a little bit more complex, or rather less reliable. But my focus is mostly on biogeography. And the Atyidae, they are present in all continents except for Antarctica. The highest diversity, however, is in the Indo-Pacific, and it is where all of the Caridella group are concentrated. This is a map of the collection from 2012 work. It was, like I said, it was very inclusive, so it gives you an idea of how this family is distributed across the globe. And this area is the area where the Caridella group is distributed. There are some Caridellans in East Africa, but the Atlantic or Europe or the Americas, there are no Caridellans. And I'd like to underline the high diversity that there is in the Indo-Australian archipelago, or rather in Australasia. However, this diversity can be an artifact of the fact that most researchers were focused in this area. And this, of course, can be evidenced by the lack of knowledge about African Atyides. On the other hand, this area is very geologically complex, and this high diversity could be a result of that. So biogeographically, it could make sense that this area has the highest diversity. So what did I do? I used a genomic approach to add data, to add confidence to the results that we're trying to achieve phylogenetically. And the approach that I decided to use was the exon capture. The exon capture is an approach that is very useful for non-model organisms because of its advantages. First of all, it does not need an annotation. It does, however, need a model annotated genome to annotate the transcriptome and design the base. What the exon capture does is basically just you sequence a transcriptome of one of your a specimen from the group that you want to work with. And then you use this transcriptome against a model genome, an annotated genome that doesn't have to be too close to your group, but has to be close enough so the same genes will appear. And then you find what from this transcriptome, you align this against the genome and find the exons. And then you produce baits to retrieve these exons from DNA. So it has the same advantages of a transcriptome-based research without its limitations. For example, it has all the markers are protein coding, which is very useful for studies involving selection, but it's also interesting for uniting different selective stories into a single one and avoiding marker biases. It also does not rely on preserving the RNA. RNA degrades within minutes once you kill the specimen. So it makes impossible to use, for example, museum specimens. So you use the RNA, you retrieve the data in the RNA from the DNA, which makes it useful for use old specimens, or even to in the collection when you don't have the access to preserving methods that will allow you to preserve RNA. So all this makes it ideal for no-model organisms. But we did something extra to this approach. So the normal approach would be sequencing the transcriptome of the specimens that you want, aligning it against an annotated genome, and then finding the exons and design the baits to retrieve these exons. We added a step, which is now is very common done for exon capture approaches. We constructed a phylogeny with these transcriptomes, and then we inferred ancestral sequences from this phylogeny, and then we use these ancestral sequences to design the baits. And I'm going to show you more visually in the results how this is done and why we did this. Once we did this, we proceeded with the phylogenetic analysis and the biogeographic analysis. So the phylogenetic analysis, I did a maximum likelihood tree with IQ tree and a Bayesian inference tree with these two. For the Bayesian inference, I used the multi-gamma substitution model, which also I will explain within the results why this was useful. And for the biogeographic analysis, oh yes, and I used the closer to calibrate. And for the biogeographic analysis, I used the Bay Area to make an ancestral range reconstruction. And I also explained why we had to use this model. So for the exon capture, in red, you see the transcriptomes that we actually sequenced and used to produce the baits. And in green is the same, but for the outer group, because we need an outer group to produce the phylogeny. And the smallest, the small one that are not marked in red and green, the small tips, these are transcriptomes that we sequenced, but we didn't produce, we didn't design baits based on them. We just used them to produce the phylogeny. So initially, we produced these many baits for these many axons for each of the samples. And then we calculated, we inferred the ancestral states through these nodes here. And we use these sequences, these ancestral sequences to produce new baits. And then we gathered everything together to actually aim for this node here with N2. So in the end, we end up with 40,000 baits and 1,300 unique genes and 8,000 exons. So with all this, we ended up with a 2 million base pair alignment. And the importance that for this approach is that because we use markers that are under selection, the p distance is very important for exon approach, for exon capture approach. The p distance is the percentage of differing sites between sequences. If the p distance is higher than 12 to 15%, the baits will be too limited, or the script will lose power to find the homologous sequences. So we needed to find the ancestral state because the ancestral state has half of the distance to the tip, between the tips. So if you use the ancestral state, instead of using one of the tips, we reduce this p distance to half of what it was. That said, I would like to point out that we had very good results for the outgroup, which was the Atyidae group. So I believe that the same baits could be used to obtain a decent alignment for the entire Atyidae family. You can see through this graph here that both nodes, even node 1, which is the node with the outgroup, and node 2, which is the node between with the in-group, have a very low p distance, and both of them are below 12%. Most of them are the mean or whatever. So this is the maximum likelihood tree, and the values on the nodes are bootstrap values. And this was done mostly as a preliminary analysis that helped us to set up the Bayesian analysis. And to comment it on more detail, we have first the outgroup here. And we had a few groups that were more or less maintained in both analyses. There's one big differing point that I will point out, but most of the groups were more or less maintained. The first group is the Atyidae group. I cannot call any group Caridina group because Caridina is in all of them. But Atyidae group contains this separate genus, which is Atyidae. And I had this other group. We have this other group that I call Sulawesi group because most of the species are from Sulawesi. Not all of them. Some of them are from other parts of Indonesia. But we have this very shallow part of this subtree is endemic to Sulawesi lakes. I want to draw your attention to these branches here and try to show you how long they are if compared to these branches. This phenomenon called heterotachy is very problematic for dating because, of course, a big premise of time calibration is that the whole tree obeys the same parameter distribution. And this is why I used a multi-gamma approach, is an algorithm for the substitution model prior that allows the gamma parameter to vary between branches. It's not the same as the relaxed molecular clock. The relaxed molecular clock allows the distribution to vary between, allows the values to vary between branches within the same distribution. The multi-gamma, they add a hyperparameter, which is the gamma distribution. You must remember, if you ever work with substitution models, you find that gamma 4 parameters or whatever, it adds one gamma parameter for each branch. And then you can actually find different rates for each branch. Continue with the tree. We have this big group here, which I call the Limnocaridina group because it contains Limnocaridina. And this small group here, which is Caridina brevicapalis group. And again, you can see that the heterotachy is very spread all over the tree. We have this group here, which I call the true Caridina group because it contains the types species for this genus, which is Caridina typus. And finally, no, not finally yet, we have the Marozina group. And again, these long branches. And finally, we have this group that I call the Paracaridina group. So the only difference between the maximum likelihood and the Bayesian inference is in the positioning of the true Caridina group. The true Caridina group here is sister to the Marozina group. And you can see that the bootstrap between them is very low. It's 14, if I'm not mistaken. And in the Bayesian tree, the true Caridina group is sister to the Lancaris group, which is the most external tip within the Paracaridina group. So the Paracaridina, I consider it a smaller group, and then Lancaris as part of the true Caridina group. Before I continue, I would like to show you, if you all, I don't know what you work with, but this is very interesting. These, the red one shows the tree height prior, the trace for the tree height prior with a single gamma approach, and the blue one shows with a multi-gamma approach. So you can see that with the multi-gamma approach, the dates, the tree height varied much less. So this added a lot more confidence to the intervals. The intervals didn't vary so much, and the tree height doesn't. The tree height with the single single gamma approach, when there is heterotachy, they tend to be higher. You tend to get older dates, and here we not only get more recent dates, but we also get a narrower confidence interval. Finally, we have the final results. I made this big picture here. This picture has a lot of information. So I'm going to go through it just to explain the information, and then I'm going to try to make it more palatable within the story. So first of all, we have the regions, the biogeographically relevant regions that are considered. They are marked as letters of the alphabet and represented in the matrix and with the colors on the right. So each tip distribution is represented on the matrix, and on the nodes, you can see the distribution of the ancestor, covered by the ancestor, for the ancestor. The red branches are the amphigenous taxa, and the black branches are the landlocked taxa. And this is important because biogeographically, we want to know how they got where they are. To infer the ancestral state, if it was amphigenous or landlocked, I considered the reconstructed ancestral area if it was separated by ocean or not. Pardon me. And for a comparison, I use the Mesquite program to map these two characters with a likelihood algorithm along the tree. So there are some nodes you can see below here that are marked with an asterisk, and these nodes represent where the Mesquite and the ancestral range reconstruction disagree for the ancestral state, for the life history, if it was amphigenous or landlocked. You also see arrows in some of the nodes. These arrows point out the nodes that have posterior probability below 0.7, but posterior probability for all other nodes are above 0.7. So first of all, we have the Atyidae group again. The Atyidae group is mostly amphigenous. It has one landlocked species with a very long branch, and this species is problematic. It doesn't make sense that's there. This is one of the problems that we have that we are trying to sort out, because we could not find signs of contamination, but it doesn't make sense that this species falls within this group. So we think that what might have happened is misidentification, but we don't have the specimen anymore. So this was a problem. So we're gonna sequence another Caridina EV and see if it falls in the same place. And if it falls in the same place, then we have to deal with it. And if it doesn't, then we have to deal with this because we don't have the specimen anymore. Then we go to the Limnocaridina group, and this clade is a bit more interesting because the landlocked species, they arose multiple times along this subtree. And you can see there's a lot of asterisks because the ancestral state for amphigenous or landlocked is not agreed upon. The ancestral range reconstruction between the ancestral range reconstruction and the Mesquite approach. Another thing that's very interesting here is that there are the appearances of the origin of new landlocked species have an interesting pattern. First, there is this Edoneus here, which is completely different genus. So maybe it involved a long lineage, or maybe we don't have the fossils, or we don't have all the species that show that, but maybe it shows particularly evolutionary history that that's involved with this change. Then we have Caridina thomasi here, which makes more or less sense, is a bit more restricted within the range in this group, in the sister group. Then we have two clades that are completely landlocked. One is an Asian clade that contains Indian and Middle Eastern species, with Caridina chawani, Caridina sharazade, and Caridina hodgarti. And we had an African clade that contained two Eastern African species and one Western African species. And this is kind of tricky because they are landlocked. So, and there is no connection between the basins. So this is kind of tricky, and it doesn't make a lot of sense at first sight. And we have another African group that is landlocked, involving Caridina nilotica and Spatulirostris, which also is kind of tricky because Caridina Spatulirostris is endemic to Mauritius, and Caridina nilotica is related to a species complex, and it's in Eastern Africa. Caridina fijiana and Caridina macrophora, they also make a little bit more sense because they are in a more restricted area than the sister group. So it seems like they got isolated in some place, and then they became landlocked, they evolved into landlocked species. Interestingly, there is this sister to there is another landlocked species here, that's Caridina palawanensis, that's isolated from everything. The Caridina palawanensis is within a species complex. You can see there's three Caridina gracilites here, and they are not monophyletic. We suspected that Caridina gracilites was a paraphyletic species complex, and we decided to include representatives from multiple populations. And this pattern could be just a matter of species delimitation. It could show it's a matter for a much closer look than we have here. We have to actually sample and do a phylogeography of the entire species complex, like Valentin has been doing with other species complexes within this family. But this highlights the problem, both taxonomy and with the morphological definitions, the morphological characters that we are using, and with the understanding of the evolutionary history in this family. Then further in the tree, we have the Sulawesi clade. It's almost totally landlocked with one exception, that's Caridina propinqua. And this is very interesting because Caridina propinqua is a species complex. There's no doubt of that. And the boundaries are very ill-defined. Because it's even being a species complex, the fact that it is amphigerous, it shows a reversal that it's the only reversal in the tree, and it's kind of unexpected. My first thought was that maybe it's not landlocked, because a lot of species are defined as landlocked because of the size of the egg, but this is not always true. Like Limnocaridina has a amphigerous egg, but it is landlocked, and this could be the case. But Caridina propinqua specifically is one of the very few species that actually has lab experiments on their development to show that they are actually amphigerous by all means. So this is an interesting, and this is something that we cannot explain as of now. Further in the tree, we have this, before that, The split of the Sulawesi clade occurred exactly on the KPG boundary. And it's interesting to note that the whole scenario is that we had an amphigenous ancestor splitting into an amphigenous sister group and a landlocked specimen here that diversified within Indonesia and mostly Sulawesi. The amphigenous clade presents an ancestral range. If you can go here, the ancestral range of the amphigenous clade includes, pardon me, the Southeast Asia and India as well. Whereas the ancestral range for the Sulawesi group includes mostly Southeast Asia. This is interesting because India was very, very stable during the KPG boundary. And it was obviously volcanically active, but the environment was stable and the sea levels were lower. And this could have provided both the salt water for the larval development and the fresh water for the adults. Whereas in the Southeast Asia, where Sulawesi was beginning to emerge at this point, it was a much more active and much more unstable environment. And this could have locked these species within a single basin and turned them into landlocked species, into landlocked clade. Further in the tree, we have the Brevicapalis group, which is a very small group, also involving a species complex with Caridina Brevicapalis. It's interesting to note that this event that presents a major split in the Paleocene boundary, which is also shown by other amphigenous, majorly amphigenous clade. Like you have the major split, the origin here of the ancestor within the Caridina Brevicapalis group is in the Paleocene-Eocene boundary. So is the Atyidae group here, between Atyidae and the other Caridina sister group to them, and also in the origin of this small group, and also further within the true Caridina group. So we have this Pliocene-Eocene boundary that seems to have some relevance for amphigenous clades. And then we have Marusina and Parisia. And we see that the distance, the branch between Marusina and Parisia and the Caridina sister group is very long. And this could be an effect of heterotachy, could be incomplete lineage sorting, and it could be it could explain why why taxonomists felt inclined to describe new genera here. But Parisia is a very messy genus, and it seems to come from pure reckless taxonomy. So I won't comment further on that. It has a very long branch, and these two genera are landlocked, in contrast with the Caridina that are involved, that are amphigenous. The Caridina groups that are sister to these two genera are amphigenous. So there is some evolutionary history there. Probably, if we check more closely this group, including these two genera, we'll find some interesting story there, but it's much more local than what I'm searching for. Then we have the Paracaridina group, and you show again the Paracaridina is also paraphyletic. We had a lot of small Caridina groups within the Paracaridina, and the Paracaridina group is purely landlocked as well. And finally, we have the true Caridina group, and you can see that here is sister to Lancaris, and you can see also that there is no arrow in this node here, so the posterior support is much higher. That's why I considered this relationship for the main tree and not the maximum likelihood, not what the maximum likelihood tree recovered. Within the true Caridina group, we have Caridina typus diverging from its sister groups in the Miocene, which suggests that it's very wide distribution. You can see in the matrix, almost all of the squares are colored for Caridina typus. It was achieved recently. So this was very tiring for me, so I cannot imagine how boring it was to you go through the tree just reading what's shown. So I would like to show you a more dynamic version of what this history I'm trying to tell here. So the story of the Atyidae begins at 95 million years ago, when this group originated. The ancestor was an amphibian species, and the ancestral range reconstruction includes most of the Indo-Pacific. The Indo-Pacific, as we know, however, did not exist yet. So the distribution could be something around the Indian Ocean as a whole. It could also be a Gondwana taxon, distributed further to the east through Australia and Antarctica, but also a Eurasian taxon centered in China. But it could be spread further to the west towards Europe, but that would depend on the reconstruction of the sister group. Then we move to 80 million years ago, when the major groups began diversifying. The ancestral range was more focused on Southeast Asia and Pacific. At 75 million years ago, the Marozina and Caridina verticalis groups diverged in a more northern region. And this is interesting because the Indo-Australian archipelago was very incipient at this point, but the major land masses such as Palawan, Borneo, Taiwan, and even Japan were already forming. But they were much more to the north of their current position. So this could explain this, inclusive reconstruction. At the Cretaceous-Paleogene boundary, we had the divergence of a majorly landlocked clade, the Sulawesi clade, and Sulawesi was just emerging at this point and much more connected to Sunderland. Both were central to the ancestral distribution of this clade, so this would explain the transforming landscape could explain how it changed through the Cenozoic. While this population diversified there, its sister population maintained its amphibian state and was, in India, as I explained, it was much more stable through the Cretaceous-Paleogene extinction. Then at 55 million years ago, we have the Paleocene-Eocene thermal maximum, which was a hyperthermal event that caused the precipitation to increase and the oceanic levels to quickly oscillate between 50 and 150 meters above from today's level. The salinity dropped drastically in all oceans, and at this time, we see multiple widely distributed amphibian clades suffering major splits, two of which gave birth to landlocked taxa in India and Africa. Not much later, from 50 million years on, the contact between Australia and the Southern Shelf intensified, and the Indo-Australian archipelago transformation began to accelerate. At this time, we see the speciation rate in the Sulawesi clade increase, so we see shorter branches along the Sulawesi clade. Between 40 and 45 million years ago, we see the divergence of a landlocked Asian clade with Indian and Middle Eastern species. At this time, the extremely high sea level from the thermal maximum was still falling, and the shallow Parathetic Sea extending from Europe to China turned the Central Asia into an amply connected archipelago. So you can see in the map, there's a bunch of islands, and these islands could be isolated following the lowering of the sea level and the uprise of the Himalayas. Then it would provide the opportunity for the diverging the Indian and the Middle Eastern species. And then we see the diversification of amphibians and landlocked locally and amphibians all over. And we have at 20 million years ago, the East Africa rifting. The eastern and western basins in Africa, they used to be very connected. When the land masses rose in the east during the East African rifting, the Central East basins that used to drain in the Indian Ocean were captured by Atlantic drainages. And the dates of these events are in complete agreement with the separation between Caridina togoensis and the other East African species. Then we move on in the Miocene to 15 million years ago. And then we had a major oceanic change between 15 and 4 million years ago. It's called the Miocene Indian Ocean Equatorial Jet. That was a very strong westward current that was formed with the narrowing of the passage between the Indian and Pacific Oceans. And in accordance to these dates, we have the split between Caridina nilotica and Spatulirostris. So Caridina spatulirostris was isolated in the Mauritius Islands, and the ancestors proceeded to Africa to give birth to Caridina nilotica. And we have Caridina typus spreading all over Indian Ocean. They split between Caridina typus and its sister species was about in this time as well. And then we get to the present with a very massive family, a bunch of badly described species, and a lot of evolutionary history that we actually don't know. And with this, I finish my presentation.